Hello everybody, welcome to Knowledge Without Bar. Today we will discuss about different types of bioreactors. In the previous tutorial, we have discussed about structure of the bioreactors and how does the bioreactor work. So if anybody missed the previous tutorial, you can see this using the link I have provided in the description box. So let's start the session. Types of bioreactors. There are several different types of bioreactors available at present in the market according to their requirement in the industrial scale as well as product development. But there are 8 different types of bioreactors which are very essentially used in different industries. So what are those? Number 1 is start tank bioreactors. Number 2 is continuous start bioreactors. Number 3 is air-lived bioreactors. Number 4 is bubble column fermenter or bubble column bioreactor. Number 5 is packed bed bioreactors. Number 6 membrane bioreactors. Number 7 fluidized bed bioreactors, and number 8 is photo bioreactors. So before I discuss in detail this 1 and 2, the start tank bioreactors and continuous start bioreactors are similar in nature. There are some differences that we will discuss shortly, but they are structurally very much similar. At the same time, the air-lived bioreactors and bubble column bioreactors, these are also structurally very much similar. And these 5, 6, 7 and 8, they have separate structural integrity. So start tank bioreactors and continuous start bioreactors, for the starting or the circulation of the media, we use the impellers. That the function of the impellers I have already discussed in my previous tutorial. And in the air-lived bioreactors and bubble column fermenter, bubble column bioreactor, we use the air or spurger for the circulation of the media. So how they function? Let's see. So start tank bioreactor, they consist of a cylindrical vessel with an agitator or impeller that stirs the contents, ensuring uniform mixing and distribution of the nutrients and gases. And start tank bioreactors are versatile in nature and suitable for a wide range of microbial cultures, including bacteria, fungi, and yeast. So the start tank bioreactors have a cylindrical vessel with a impeller, and this impeller circulates, and for that reason the media present within the bioreactors are also circulated. Now the similar principle is used in the continuous start reactors or continuous start bioreactors. So what is the difference between start tank bioreactors and continuous start bioreactors? The difference is, in case of the start tank bioreactors, which is also called the batch start tank reactors or BSTR. So in this case, a fixed volume of media is used for the culture. So there is no chance of mixing a fresh media during the reaction. So after completion of the reaction, the entire media utilized in the BSTR or STR are recovered and the products are also recovered. So this is very much simple, very much similar to the batch culture that you all know, batch culture and continuous culture. In case of the batch culture, the fixed volume of media is used from the beginning to the end of the reaction. So in case of the STR or BSTR, which is very similar to the batch culture, where all 4 stages of the bacterial growth have been seen. So what are those? Lag phase, log phase, stationary phase, and death phase. All these 4 stages are seen in the STR. But in case of the continuous start reactors, what happened? The substrate or media are added after some time, and at the same time similar volume of the used media or product is recovered from the tank. For that reason, the volume within the tank remain fixed, but a continuous supply of the fresh media taken place within the reactor, and that reason all the microbes present within the bioreactors have shown the log phase because they are continuously dividing and there is no nutrient limiting factor. So this is the basic difference between starting bioreactors and continuous start bioreactors. Number 3 is air-lived bioreactor. As I already told that air-lived bioreactors and bubble column bioreactors are utilized the spargers or the air bubbles for the stirring of the media within the bioreactors. So what are the function of the sparger? Again, I have already discussed in my previous tutorial. So if anybody missed, you can see this from the playlist. So air-lit bioreactors are characterized by the use of the gas sparging to create a circulation within the reactor. They typically consist of two interconnected zones, a riser and a downcomer. So these are the two zones. This is the riser zone, the white colored region, this is called the riser zone, and outside the blue colored region, this is called the downcomer zone. So gas bubbles are injected into the riser section, creating buoyancy, causing the mixing and circulation of the culture. How? So this is the sparger from where the air bubbles are comes out into the rising section. So like this, the air bubbles are comes out from the sparger in the rising section. It goes up, and when it comes in the downcomer region, it circulates like this. So it produces a circulating motion within the bioreactor. And as the air bubbles causing the circulating motion, within the bioreactors, the media are also circulating. So instead of using any impeller or agitator, we are still creating the circulative force by using simply the sparger. So in which cases the sparger based circulation are used, that is the air-lit reactors are used. This is specifically used where shear sensitive cultures are utilized for the bioreaction. Shear sensitive means it particularly for the fungal pathogen where the mycelial forms are present. So you all know the fungi produce the fungal hypha produces the mycelial mat. So this mycelial mat, if we use this impellers for the agitation, then there will be every chance of entangling the fungal hypha with the impellers, and shearing of the fungal hyphae takes place. That will limit the growth, will never found the optimum growth of the fungi within the bioreactor. So in those cases where the shearing of the fungal hyphae or shearing of the microbes are essential factors, in those cases we generally use air-lived bioreactors or the bubble column bioreactors. So similarly, as just like the air-lived bioreactor, bubble column fermenter or bubble column bioreactor also have a sparger, but the only difference is in case of the air-lit bioreactor there was two separate chambers. One is called the riser, another is called the downcomer. But in case of the bubble column, there is no separate two chambers. The single chamber bioreactors where the air bubbles are comes out from the sparger and it causes a random movement within the bioreactor, and this random movement of the air bubble causing a random circulative force within the sparger. So bubble column fermenter or bubble column bioreactor is a simplest form of air-lived bioreactors. And another thing is that at present in several cases air-lived bioreactors are very much popular for the industrial purposes. Now packed bed bioreactors. So as the name implies, packed bed means here the microbial cells are packed into a solid surface. For example, beads or fibrous matrices, and we immobilize the microbial cells within those beads or within those matrix, and rest of the region of the bioreactor are kept free. So within these in the media, this microbial cell definitely can produce the products, and it will releases, and it will be easy to recover the products from the bioreactor. So where the packed bed bioreactors are used, it is principally used in case of the production of the enzymes, because the enzymes are very much liable to destruction. So if we are using the normal bioreactors where the products and microbial cells are mixed with one another, then during the recovery stage, it will undergo a long processing, and this long processing, because we need to separate out the microbial cells from the product. So during those long processing hours, the enzymes may degrade. For that reason, in those cases, the sensitive enzymes, sensitive molecules, for those cases we use the immobilized beds of microbes, which is called a packed bed bioreactors. The membrane bioreactors are also very similar with the packed bed, but in these cases, the microbial cells are separated from their product by a semi-permeable membrane. As it is a semi-permeable membrane, the microbes cannot passes through this membrane, but the products produced by these microbes can easily passes through this membrane and accumulated within this separate chamber. So it will be also easy to recover the product, because microbial cells are always separated from the product. And this microbial cells, if we change the culture media with the fresh nutrients, then it will always remain in the functional state. So membrane bioreactors are also used for the specific cases where enhanced control has been operated. Number 7 is fluidized bed bioreactors. This is very specialized bioreactors where a stream of gas or liquid is used to suspend the solid particles, creating a fluidized bed. So the large gas bubbles are released from the bed of the or lower portion of the bioreactor. For that reason, it causes a fluidic nature within the bioreactor media, and the microorganism can attach to the solid particles or form a biofilm, allowing for continuous cultural growth. And fluidized bed bioreactors are advantageous for high density cell cultures where the cell density is very much high within the bioreactor, and it is specifically used in the wastewater treatment and production of the biofuels. And another case is that where the target products are of different, consisting of different masses. So when we fluidize the base, so in that cases definitely the lighter polymer catalyst will travel much faster, and it will accumulated in the top of the bioreactor, and the heavier polymer catalyst, due to their heavy mass, they cannot travel that much faster, and that will separated from the lighter polymer and settle down towards the lower portion of the bioreactor. So it will be easy to segregate out these two type of polymer catalyst after the reaction. So in such cases where different types of products are released and we have to segregate out the product or the end product of the reaction, in such cases the fluidized bed bioreactors are used. And in case of the wastewater treatment and sewage cultural system and the production of the biofuels, the cell volumes are very much high, and those cases after the reaction, if we try to segregate out the materials from the this high volume material, it will be a very tiredest job. So for that reason, this fluidized bed is used where it will be easier to segregate out this polymers after the reaction. And number 8 is photobioreactors. So photobioreactors are specialized bioreactor designed for the cultivation of photosynthetic microorganisms such as algae, cyanobacteria, etc. and they provide the controlled light exposure and are often constructed as transparent vessels or tubes to maximize light penetration. One important thing is that during my first tutorial I told that both the glass and steel materials are used to produce the bioreactors, but in case of the photobioreactors, as the organisms are photosynthetic, so they needed the light. For that reason, we cannot use steel material for their preparation, so we have to go for the glass bioreactors because light is essential for the photobioreactors. And photobioreactors are employed in the production of biofuels, high value compound, and wastewater treatment. Usually several parallel chambers of uh, glasses are utilized for the production because this will generate the high biomass in a particular period of time, and some filters are used to segregate out their products from the cell culture. And these photobioreactors have a very complex mechanism and structural mechanism, and they have varied utility. So we can discuss separately about different types of photobioreactors, how they made up, how they have the how much they have the complexity, and what are the different types of the application of these photobioreactors. We can discuss it separately. So till then, thank you for listening, and please subscribe the channel for getting such more videos in future. Thank you.